Right, we're looking at blood, the topic blood. We'll have non-blood questions in, but let's run with it. So the word transgenic, gene and movement. Right, it's when you have the DNA or genes from another species. Yeah, so what would be an example? There's a human and would you do that? Yeah, I suppose you could get a gene from a sheep, put it into human, and then he'll become more sheepish. Da-da. No, I don't know. So let's move on. Transgenic sheep can be reproduced by cloning. What are the advantages of reproducing transgenic sheep by cloning? Right, if you've got the genetically identical, so if you've got the correct genes that you want or the features, you can just keep reproducing them. Genetically identical. They are genetically identical. So with this sheep, you've got a clotting factor X. It's not in this question, but it's earlier. So you've got human clotting agent X, which you've put into the sheep, and then you can harvest it afterwards. So it's genetically identical. 2, you can produce the clotting agent X without having to use human blood. You can produce lots of them, produce lots of sheep with this clotting agent. You can also you only have to genetically, right, let's genetically modify the sheep once, the organism or the sheep in this case once, and then you can just keep using that sheep to produce more. So what are the advantages? They're genetically identical. They produce, and you can harvest the clotting agent. You can produce lots of sheep, and you only have to genetically modify once. And once you've got the genes that you need, you can stop. And I suppose the other advantage is, what's the other advantage? There will be no shortage. You'll have plenty of supply, which is in effect that. Yeah. Name the small structures. Name the small structures in normal plasma that involved in blood clotting. They are called platelets. Platelets. Why is it important to have blood that clots? Right. First of all, it stops a patient or person losing lots of blood. That can be bad. And once it's clotted, it stops pathogens entering. So it forms like a barrier and causing disease or infection. Right. The table gives statements about blood cells. Some apply to red blood cells and some to white blood cells. Remember what we said, you've got a red blood cell and you've got a white blood cell. There's 2 types of white blood cells. You have the lymphocytes you have the phagocyte. Red blood cells are this shape. They have a biconcave shape. Why? Because it increases the surface area. Right. There is no nucleus. Why is there no nucleus? Because then there's more room for hemoglobin or globin. And that helps transport. What does hemoglobin do? It helps transport oxygen around the body. Right. Right. Contain a nucleus. They do not contain a nucleus. They do. That does transport blood. Right. Okay. Produces antibodies. Red blood. They don't. They do. So the lymphocyte produces antibodies. The phagocytes engulfs pathogens. Right. Is it biconcave shape? Yes, it is. Is that? No. Does it ingest pathogens? No. Does that ingest pathogens? Yes. Eats. It eats pathogens. What did we say pathogen was? Pathogen is a disease causing organism. Oh my God. Did anybody even hear me? Now you can hear me. That's fair enough. You can hear me now. Number may increase numbers, right? Not with red blood cells, but definitely with, and these are called, some of them become what we call memory cells, and they are hang around the blood in the body, in the blood, giving you immunity. Right. Oh, let's go up. Let's have a look. Right. Okay. There you go. Some athletes preparing for long distance race at high altitude. So your body produces an increasing number of red blood cells. And the red blood cells return when they return back, it remains high. So you've got more red blood cells. Why is it an advantage in long distance races? So first of all, red blood cells have or contain hemoglobin. So let's just say you have more hemoglobin, which means you can transport more oxygen to the muscles for aerobic respiration. So for aerobic respiration, what happens with aerobic respiration? Right, so what happens? So that means more energy is released for the muscles, and you have less lactic acid building up in the cells, in muscle cells, muscle cells. Right, so that's what happens. So that means that you can run, so you can run further and faster because you've got enough oxygen. Right, so what happens? Look, there's a standard answer to this, right? So you've got red blood cells, they have more hemoglobin. If you have more hemoglobin, you can transport more oxygen to the muscles for what? Aerobic respiration. That means you've got more energy being released for the muscles. At the same time, there's less lactic acid buildup in the muscles, so that means you can run further and faster. There you go. Just learn it that way. Right, with this one, if you do a 100-meter race, it takes a short time. So any respiration is done, because it's a short race, any respiration is done anaerobically. Right, short race respiration is done aerobically, and no oxygen is needed. Right, that's it. 2 marker that was. Right. The immune system responds to an infection with white blood cell, a phagocyte. So remember what we said, there's 2 types of white blood cell. Phagocyte, they mentioned here, and a lymphocyte. We're interested in this. So look, what you've got to do, it's larger, but you have to draw a lobed nucleus. This is your nucleus. Normally a nucleus is like this in a normal cell, right? But this one is lobed because when phagocytosis is being carried out, it's useful to have that. Okay, 3 marks. So you've got cell surface membrane, you've got cytoplasm, and you've got a nucleus, and it's lobed. It's that shape. It's like a cashew nut. You would have mitochondria, and ribosomes. Okay. So the key thing is, look, with this, you've got a cell membrane, you've got a lobed nucleus, and you've got cytoplasm. Right. Give one way in the structure of this cell differs from a red blood cell, and the most obvious one, right, is it has the red blood cell has no nucleus. Right, okay, now this is again a standard answer, right? What you do is you split up the white blood cells into 2 parts. Let's focus on the lymphocyte. Lymphocytes produce antibodies. So what happens is, look, there's a bug there. There's your white blood cells. It's got a little marker, an antigen. So what happens is this lymphocyte here, these are lymphocytes. right? They produce antibodies. So look, this little marker here is called an antigen, and the lymphocytes produces an antibody. That then goes over and attaches itself there. Now it can either kill that, get it to explode. It can act as a marker. It can mark it, so then when the phagocyte comes, it knows what to eat. And it can also get them to stick together. So when there's lot of these pathogens stuck together, they can't run off. The phagocytes can eat them. Right. So the lymphocytes produce antibodies for specific antigens. Then what happens? Lymphocytes produce antibodies for specific antigens. Then what happens? Right. Some lymphocytes become, what do they become? Memory cells, and stay in the blood next time the pathogen comes along. Let's use the word pathogen. Next time the pathogen invades the memory cells produce lots of antibodies and quicker. This is called a secondary immune response. Right, let's move on to the phagocytes. The phagocytes engulf and release digestive enzymes. That means they just eat them and then throw away the waste. Release digestive enzymes and kill the pathogens. Okay, let's move on. Right. Now what we need to do is draw this. Oh, we've got the axis, good. So it goes from 0 to 3000. So, okay, we can do 1000, 2000 and 3000. Right, we now need to label that. It's called altitude in meters. So I'm just going to highlight this. Just bear with me a second. It's we're going to label this x-axis as altitude, and then you have to put the units in meters. Double check that. Yep. And that is average mass of hemoglobin in grams per liter. So this in grams per liter. So average mass of hemoglobin. Right, what's the scale? It goes from 121 to 140. So what would happen if we did 121? So if that is 121, if that's 120, 121 to 140. So 20, 30, 40. Would that be sensible? Don't know about that. 120. Right, what if we did 120 here? Right, got to get the scale right because you want to use up a lot of it. Over half, 120. Okay, 120, 122, 24, 26, 28, 128, 130, 132, 34, 136, 138, 140, 142, 20, 22, 24, 144 then, and 148. So that would be 132 and 124. Right, okay, we've got that. Now we just need to draw it. So at 0 altitude is 121. So let's do that. Let's use that color pen. At 121, 122, 121 is here at 0 there. Right, the next one is 500 and 121. Oh, right, okay, 500 and 1,000 are all 121. 121. Oh, nope, I've drawn it the wrong way around. That's a bit. Right, what I'm going to do is swap these two around. So I'm going to transfer that down and transfer that up. Right, okay, so we've done that. So we had at 0 it was 121. At 500 it was 121, and at 1,000 it was 121. So it remains the same at those altitudes. Then at 1,500 it's 125. At 1,500 it's 125. 125 is there. 2,000 is 128. So you go to 2,000 and you go to 128. So that's 28 there. 2,500 is 133. 32, 33. And then finally it goes to 3,000, it goes to 140. At 3,000 it goes to 140. 32, 34, 36, 38, 40. Okay, so now somehow, I think there's a shape here. So I'm going to pretend that these are straight lines. Yep, and then I'm going to pretend that's a straight line. Oh, okay. We'll have to run with that. Right, these are straight lines with the ruler. Right, so we've got the axes with the right way around this time. You've labeled them. You've used more than half. Do not squash like this. Don't be tempted to squash it all in this half. They like you to use at least half. So then that becomes, you've got to just think about how you're going to spread it out. Use the graph to describe how altitude affects. Right, so up to there's no change, and from there it just goes up. So we can write that down. We can say up to an altitude of 1,000 meters, there is no change in hemoglobin levels. Hemoglobin levels. Then from 1,000 meters to 3,000 meters, the hemoglobin levels rise. That's it. It's 2 marks, so it's nothing. Right. So why is it an advantage for long distance runners? Compared to a person who runs at normal sea level. So you have more hemoglobin, which means that more oxygen. Right, why is there more hemoglobin? Because there are more red blood cells in the body or in the blood. So then you've got more hemoglobin, which means that there's more oxygen transported for what? For aerobic respiration. Get these keywords in. What does that mean? So that means that there's more energy released for muscles during the race. That means that there's less anaerobic respiration, so less lactic acid, so you can run further without fatigue. Okay, let's keep going. Okay, that's 27 minutes already. Right, okay. Let's just get this finished and then we can do another one. Scientists obtain blood samples from a vein. Why do you obtain it from a vein rather than an artery? First of all, it's closer to the surface. It has a thinner wall, so there's less damage and less bruising, and it's easier to recover. Thinner wall, and it has a larger lumen, so closer to the surface, so you can see it. So it can be seen and obtained. Yeah, you're not guessing. Larger lumen, so easier to use the needle to obtain blood. To obtain blood or to get blood. Obtain blood. So any one of these. So there's 1, 2, 3. Any one of those, right? So just one reason scientists would not use blood sample from a capillary, right? Capillaries are way too small to obtain blood. So just why the needle should be sterile, right? It's to stop pathogens getting into the body and causing disease. Nope, causing disease. There you go. Right, what we'll do is that's half an hour on that, so we'll stop that and we'll continue on in a bit.